Now, animal cell organism all over. Today we will only discuss about plant cell organisms and the plant cell. We got your take out your notebook. Take out your notebook and draw this diagram first.
Ini dah ada yang
What is that about mitochondria?
Metabolism.
Plastics. Those are plastics.
Which
Easy to obtain by the words.
Last time I asked you to carry pencils. Okay, you do one thing. At the last page, you draw it with a pen if possible. At home also.
what are those red things? Vesicles?
Which one? This one?
Yeah.
This one? These are small lysosomes at the same time vesicles. Hmm? Keep drawing. we are drawing plant cell, so we have to draw two matrices. Done.
Yes, ma'am.
Now, look into the diagram. What are some of the differences that you come across between the plant cell and the animal cell? The first is this cell one, the second is this one,
Yes.
last is, and one more. Can you tell me one more?
Vacuole.
Vacuole is there, and the fourth, and the fourth.
Gamete.
The nucleus is moved towards the wall, towards the periphery. If it is the animal cell, how do we draw the nucleus? We draw the nucleus in the center of the cell because there is no vacuole in a animal cell. In case of a plant cell, there are small vacuoles also, or there may be 2-3 medium-sized vacuole. In certain, there may be one big vacuole. No? Whatever, because of the position of the vacuole, the position of the nucleus is changed. So the nucleus in a plant cell is never in the center, that's why, and always towards the wall, we say towards the periphery. So these are the major 4 differences that you will always remember between the plant and the animal cell. Let us finish with the organelles first. What are some of the special organelles now to understand? We were talking about the cell wall earlier also. Let me again repeat. What about the cell wall? What are some of the properties? If I have to talk about the cell wall is freely permeable. This is one particular property that we understand. It is freely permeable. What is it made up of? It is made up of cellulose. It is made up of cellulose. The plant cell wall is made up of cellulose. What does it provide to the plant cell? It provides rigidity or framework to the plant cells. rigidity or framework to the plant cells. Also to remember, it is the cell wall which is also having protection functions for the plant cell. Also has protection functions, also provide protection, because now that you talk about the cell wall is so resistant, it does not allow quite a number of unwanted substances. Now when I say it will allow the movement of water molecules or other different substances, of course, inside, freely permeable, but at the same time it is because of the cell wall that the cells of the plant cells easily cannot break, they easily do not get ruptured, they easily do not get damaged. Okay, it is a cell wall which protects them. So that's why we say that it also has got some protection functions. And always to remember, it is the outer wall of the plant cells. Talk about the plastids next. What about the plastids? What are their categories? What are their functions? We talk about the plastids. These plastids that we are talking about can be categorized based on what function they perform. So we talk about plastids. Plastids, I said, let us talk about the category first. Categories of plastids. The categories of plastids are, number one, we talk about chloroplast. One category of plastid, chloroplast, which produces chlorophyll. And chlorophyll can trap the solar energy. Chlorophyll will trap the solar energy. And since they are able to trap solar energy, whichever cells will be having the chloroplast, they would undergo the process of photosynthesis, and they would be capable of preparing food in the form of glucose. So this is always associated with the word chloroplast. Chloroplast, a particular type of a plastid. If a cell has chloroplast, meaning the cell would be definitely producing chlorophyll and making that plant part green in color. But what would be the function of that chlorophyll that they are going to produce? That chlorophyll very much would be helping in the process of photosynthesis, allowing the plants to prepare food. What is the second type of the plastid? That is chromoplast. And what would this chromoplast produce? They also would be producing various other pigments. They would be producing various other pigments, such as we may talk about carotenoids. We may talk about carotenoids. Carotenoids are one of the most famous, most frequently occurring pigments in the plant cells, carotenoids. And what would these carotenoids functionally bring about? Give color to plant parts. It would give color to the plant parts. So when we say it gives color to the plant parts, what do we mean? What do we understand? Give color to plant parts. It would give color to the flower, to the fruit, to the crop. I may talk about the color of the, when I talk about the chromoplast, it is the chromoplast which is responsible for the color of the papaya, color for the carrots, beetroot. Okay, so we are talking about the chromoplast. So when we look into chromoplast, so whenever we are talking about the chromoplast, remember what does the chromoplast do? It brings about, or it gives color to the plant parts, gives color to the plant parts. What about the third? We talk about the third type of plastids as leucoplast. What is the third type of plastids? Leucoplast. Now what does the leucoplast do? It does not have any coloring property, because these are also known as colorless plastids. These are colorless plastids. But they also help in, but they have a definite function, of course, and they help in storing fats. Okay, storing fats or oil droplets in any particular cell and starch. So they basically have got some storage functions. They help in storage functions. So what are the functions of the leucoplast we come across? Nothing to do with any coloring, but their functions are absolutely different set of functions. They help in certain storage functions. Next is, we talk about the vacuole. What is this vacuole containing? It is a thin-walled, bubble-like structure. It is a thin-walled, bubble-like structure. And this thin-walled, bubble-like structure contains what? It contains the water and absorbed by the roots. So what is the water and the minerals also known as? The water and the minerals also known as the plant sap. What is the plant sap? It is the water and the minerals that has been absorbed by the plant's roots and carried by the xylem. Okay, we'll study about the xylem in detail. What is the function of this vacuole? Provides turgidity. Turgidity means stiffness. Turgidity or stiffness to plant cells or plant parts. And the sap also brings about the dissolving and storing of food and salts. So it is the sap which actually, as just now I said, that sap is nothing but the water and the minerals which the roots have absorbed. So this sap, when I talk about this particular sap, would actually also help in storage. What type of storage part? Yes, food. When I talk about that the plant cells are producing glucose, yes, the glucose gets dissolved here in every cell. The glucose is not in a crystal form or anything, but the glucose inside a plant cell is always in a dissolved form. So now I talk about the glucose. The glucose is always in the dissolved form inside the sap, and in the dissolved form only it will be transported. And the excess form always stored in the plant parts. Yes.
Sap also
brings about dissolving and storing of food and salts. Storing and
sap also
brings about dissolving. So what are some of the functions of the sap to understand? If the plant cell has adequate amount of sap, the plant cell will have a shape. Remember, the vacuole in a plant cell also helps in maintaining shape of the plant cell. Now, how do we understand that the sap is responsible for the maintenance of the cell shape? I talk about two pots. In one pot, I have watered in the morning enough. And another pot, I have not watered for the last 2 days. Will there be difference in the leaves of the plant?
Yes.
In both the plants, there is a difference in the leaves. In the first plant, which I have already watered in the morning, even though there is sunlight, are they going to get completely dried out? Not much. And the other one which was not been watered for the last 2 days, they will wrinkle and dry up. But I have watered right in the morning 6 o'clock. So by the time it is 8 o'clock, before it is fully strong sun, how do you get to see? Or if I talk about early morning, how do you get to see the plants, the leaves of the plants? In the early morning, how do you get to see the leaves of the plants? Very fresh. The leaves of the plants early morning are very fresh because the sunlight is less, the loss of the water from the leaves are less, and the leaves have got so much of water. The sap is full with water, and that is making every cell tight filled with water. That is what is known as turgidity. What is the meaning of turgidity? Condition of the leaf cells which are tight filled with water. Condition of the leaf cells which are tight filled with water. If the leaf cells are tight filled with water, then only we get to understand that the leaf parts would look fresh. Then what would happen when the leaves have given away so much of water? The leaf cells. Now the vacuole in that case, will it still appear this way? No, the vacuole will not appear this way. When the leaf cells are giving away water, the vacuoles will appear like this, wrinkled. The vacuoles are getting dried out. No, wrinkled it is. So this is what you have to remember. So far we talk about the cell wall, plastids, vacuole. Now another section of the chapter that we have to discuss would be relating to concentration of the sap and the surrounding water. Now, since we are already talking about the vapor and the sap, now related to that only, we are looking into the last section of the chapter. Now, try to understand. I am talking about 3 beakers. I am talking about 3 beakers here. So, in this 3 beakers, what did I do is I took some solution. What am I saying? I am taking 3 beakers like this. I took 3 beakers. And in this 3 beakers, what did I do? I have taken some solution. So I have taken some solution. I don't know what is the strength of the solution. So what did I do? I have taken some solution here and I don't know what is the strength of the solution. But I just know that there is a solution here. Solution meaning this solution must be a solution of water and sugar. Maybe a solution of water and sugar. When I say solution meaning something is mixed with water. No? So now in this, what is happening? There are 3 beakers taken, and in each of the beakers there is a particular solution. We don't know what is the solution about, and we are asked to take some cells inside. I'm just drawing one for representation, but we have to understand that there are many cells like this. So in one of this, I talk about that. I just now said what? In each of this, we have taken cells inside. In each of these beakers, we have taken plenty of cells inside. That can be a plant cell or a group of animal cells also. Right now, since we are talking about plant cells, so I consider plant cells. So I'm saying that in each one of them, what did we take? We took enough amount of some plant cells. We took enough amount of some plant cells. So let me just draw some plant cells here so that you understand what is happening. So I'm talking about some plant cells being put here. I am drawing the condition of the plant cells. Did I draw the vehicle big or small? Quite big enough. That means the cells are having already adequate amount of water. More or less adequate amount of water is already there in the sap. And all the cells are almost in the same stage that we have taken. So what did I take in this experiment? Water or a solution, solution in 3 beakers. And then certain plant cells. We have taken certain plant cells. What are these I'm drawing? I'm drawing some plant cells. Now, after a period of some few hours, what are the observations? That is what we are going to understand. After a period of some few hours, in beaker A, we see that the cell shape, what happens to the cell shape? Let us compare now in each. What happens to the cell shape? The cell shape remains as it is, no change. In beaker number A, the cell shape remains as it is. Let us talk about the other. In beaker number B, what about the cell shape? The cell shape, I'm not writing together repeatedly. Okay, let me write in case you have a confusion. I am talking about the cell shape. What is happening to the cell shape after a particular period of time? The cell shape is getting bulged. The cell is getting bulged, swollen. The cell swells up. Swollen. So now, what about the third condition? Cell shape. That is the first thing that we understand. The cells are shrunken. Okay. So after a period of time, something around some 8, 10, 12 hours, we get to see that. What about the cell shape? What is happening here? The cell shape is, in one, the cell shape remains same, in another it swells up, and in the other, it gets shrunken. Now the condition is, what would happen? Under what condition the cell shape will remain the same? The cell shape is known to remain the same if the amount of water coming out and the amount of water getting in is almost the same. If the amount of water getting in and the amount of water coming out from a cell is almost the same, then the cell shape will remain the same. Say suppose I have a classroom of 20 students, but every time 2 going out, another 2 coming in. 2 going out, another 2 coming in. Is there any change in the number of the students of the class? No, that is what is happening in the first case. So in the first case, what is happening? Maybe some few molecules which are going out and few molecules which are coming inside the cell is almost the same. Now, when, under what condition will this type of a phenomena happen? This will happen only when the solution has the same strength. What are we comparing in this experiment? We are comparing, remember, two things. We are comparing the sap and the solution. What are we comparing in this experiment? The sap and the solution we are constantly comparing. So in the first case, what is happening? The solution, this is the solution and this is the sap. I just now told you the molecules of water that must have entered and the molecules of water that must have gone out is perhaps the same. That is the reason the cell shape did not change. Then what type of a solution we are going to name it as? Now we are more or less sure that the strength of the solution and the strength of the sap is almost the same perhaps. Now what is the meaning of strength? Strength means how much, no, strength means, when I compare any particular solution, I am comparing two solutions. And I am saying that the strength of both the solutions are same. What does that mean? When I am comparing solutions, I have to compare either of the solutions and a definite amount. I may take 1 ml of this and I take 1 ml of this. And then in this 1 ml, I am not to understand that this cell is so small. The vacuole is so small. How can we compare with the solution outside in the beaker? No. If I consider 1 ml of this and 1 ml of this, it is like that. Volume by volume compare. Volume by volume compare. So now what I'm saying, if I compare, or as the scientists have compared, that whenever they have compared one volume of this and equal volume of this, then they found that the amount of solute in the 1 volume solution of this and the 1 volume sap is equal. So 1 volume of sap and 1 volume of solution has the same amount of solute, same amount of water. If it is like this. So what did I tell you? The water, in this 1 ml, amount of water in both is same. In both this solution and sap, in this 1 ml, what about the solute, sugar and salt? That also by molecule it has to be more or less the same. Then what do you talk about such solutions as? This is known as isotonic solution. And what is the definition of isotonic solution? A solution, now you are comparing. What are you comparing? Sap and solution. A solution
the same number of water
which has same concentration of solute as well as solvent. Solvent is the water. Both you have to write down in the definition. A solution which has same amount of solute and solvent as compared to sap. Did I make it clear for you to understand? So what is this? Are we defining the cell sap? No, we are defining solution. Why in this condition the cell did not change its shape? Because the amount of water here and here is same. Amount of solute here and here is same. But are we saying that this is maybe 500 ml of water? Are we talking about that 500 ml of water and that small cell? No, 1 ml of this, 1 ml of this, volume by volume. Can I compare a 6th standard boy with 11th standard boy? No. I have to compare a 6th standard with a 6th standard, 11 with 11. No, it is like that. Come to the second one. Okay, we'll have to define all these. Acha, now, when I talk about in an isotonic solution, whenever cells are put, what would be the behavior? They will never show a change. Come to the next one now. So when I talk about the next one, what would happen in this case? After a particular period of time, after a particular period of some few hours, what did we observe? What is it observed? That the cell is swelling up. Now, when will the cell swell up? When the amount of water entering is much more than the amount of water going out. Maybe for every 5 molecules of water entering, only 1 molecule of water is going out. But under what condition will that happen? When again I compare this 1 ml is to 1 ml, 1 ml of sap, 1 ml of solution, when I compare the amount of, now this is the solution I'm saying, no, this is the solution I said, this is the sap. In this 1 ml of solution, water is more. In this 1 ml of solution, water is more. Since water is more, in this sap, water is less. In this 1 ml, water is less, solute is more.
Yes, ma'am.
And in this 1 ml, water is more and solute is less. So water will always move from a region of high concentration to a region of low concentration. Diffusion example definitions you have come across. Almost same like diffusion, we are now talking about osmosis. But now that we are talking about osmosis happening with the concept of entering inside the cell, to be very specific, we should say endoosmosis is happening. Whether the cell is gaining or losing, that is what we try to understand. Under this condition, is the cell gaining water? Yes, so much it is gaining. So it is gathering 5 molecules, but hardly 1 coming out. It is gathering 10 molecules, hardly 3 coming, going out. So it is gaining more than what it is giving out. What process? Endoosmosis. Then what would be the nature of the solution here?
Hypotonic.
Hypo. Hypo means less. Less than what we are comparing. Hypo means less concentrated or dilute as compared to the cell side. So if this continues quite a number of times, if the cells or the certain type of plant or animal tissues are kept in a dilute solution for a longer period of time, sometimes we also come across the tissues start swelling, and they may burst also. They may burst also, but not so very easily. I told you, cell wall protects them. Then what about the third one? In this condition we come across, when I talk about the 1 ml of sap and the 1 ml of solution,
This condition is what?
here what is happening? The third condition was, after a period of time, some again, 8 to 10 hours, what did we see? The cell got shrunken. Why? But it will shrink only when the cell has given away too much and it has gained little. What it has given away is much more. It is giving away 8 molecules, but it is getting back only 2. Giving away 6, getting back 1. Giving away 10, getting back 3 or 4. So if that is the situation, then only the cell will shrink, no? Slowly it will start shrinking. Then the cell is giving away water by a process almost like diffusion. The water is moving from a region of high to a region of low. Now the region of high is here inside. Earlier the region of high was here. Higher water concentration, no? So now what type of osmosis we are talking about? Exoosmosis. And what type of solution? The solution will be termed as hypertonic. Understood? So only when a cell is placed in a hypertonic solution, the cell always tries to shrink, and sometimes the cell may dry out also. Same like I talk about that potted plant, I was giving you an example. I have a potted plant which I have not watered for the last 7 days. I went out for a holiday. When I came back from my holiday, am I going to see the plants fresh? No, possible that if it is a small plant, it will die out. What happened? The cells were constantly losing water, and ultimately they died out. So if the cell is losing too much water, then what it is gaining? The process is exosmosis. And if the cell is kept in a solution to experiment that, such type of a solution will be termed as hyper. The solution is dilute than the cell, set of the cell. And the solution gives water, the cell gains water. What type of a solution? Hypotonic. And the cell solution and the cell has got equal amount of water and solute. What do we consider such type of a solution as? Isotonic. Now we will write down this definition: endoosmosis, exosmosis, their differences. Write down the first question. Write differences between endoosmosis and exosmosis. Make a table. Difference between, write fast, we don't have much time. Endoosmosis and exoosmosis. Difference between endoosmosis and exoosmosis. Make a table. Did you understand this, all of you?
Yes,
The
ma'am.
concept of hypertonic, hypotonic,
Yes, ma'am.
osmosis, endo, exo. Okay, now first you write endoosmosis and exoosmosis, make a table, write on the first one. Endoosmosis, write on the first point. Diffusion of water, diffusion of water,
We have to make one table.
huh?
We have to make one
Make a table, yes. Diffusion of, or endoosmosis, huh? The first point. Diffusion of water, diffusion of water inside the cell. Diffusion of water inside the cell. Cell taking it is. It is known as when the cell takes in water. Diffusion of water inside the cell. Diffusion of water inside the cell. Exosmosis. Diffusion of water, or complete the sentence, diffusion of water inside the cell from the surrounding. We are comparing from the surrounding. Whatever with the surrounding. From the surrounding.
inside the cell.
From the surrounding.
Next one.
That is endo. Now you write for exoosmosis. Diffusion of water from the cell. Exo. Diffusion of water from the cell to the surrounding. Exo. Diffusion of water from the cell to the surrounding. That is exoosmosis, right? Full stop. Write down the second difference. Under endoosmosis, under that column, second difference. The cells become turgid. The cells become turgid and swell up. Cells become turgid and swell up. Other side, exo. Cells become flaccid and shrink. Cells become
Turgid and
turgid. Cells become turgid and swell up. For exo, cells become flaccid. Flaccid means wrinkled. The cells become flaccid and shrink. Shrink. Third point, endoosmosis. Third point for endoosmosis takes place when cells are placed in hypotonic solution. Takes place when cells are placed in hypotonic solution. Hypotonic solution. Next, other side, exoosmosis takes place when cells are placed in hypertonic solution. Takes place when cells are placed in hypertonic solution. Takes place when cells are placed in hypertonic solution. Done? Got those? Now we will write down the definitions of these three types of solutions. Write down the first one, isotonic solution. How to define? This answer is done. Three differences between exo and endo. Now, isotonic solution. Right. Isotonic solution.
What isotonic?
Isotonic solution. How will you define? What will you?
differences between
Exo and endoosmosis. Why are you writing that? We are already finished. You are in half sleep.
You told another one.
Now definitions to write for each of those three terms: isotonic, hypotonic, hypertonic. And what have you written from school?
310.
310. What do you do after that? So immediately change your clothes, have your food, and at least if possible before you come, take a nap of 15-20 minutes. Because you are tired right from the morning, and now that you come here, you are half in sleep. No? So if that happens, it is a waste of time.
I was writing 3 differences between isotonic and.
अभी वो हो गया, अभी तो वो हिस्ट्री है, वो खत्म हो गया, वो ऑलरेडी लिखा ना तुमने 3 differences.
Naturally, solution have the same amount of water molecules and the
There's
solute.
a, if you write this one, no teacher will give you. It's a format होता है. In that format you have to write. But you will understand. Mugging नहीं करोगे, wall देख के, fan देख के, mugging करोगे तो कुछ भी याद नहीं आएगा. So isotonic solution. What are you supposed to write down? Isotonic solution. The solution which has same remember, isotonic, hypertonic, hypotonic is a relative term. Relative किसको बोलते हैं? Whenever we are comparing, which has equal concentration of solute and water. Equal concentration of solute and water. Equal concentration of solute and water as compared to a cell. Equal concentration of solute and water as compared to a cell. So if you write down the definition like this: isotonic solution, a type of a solution which has equal concentration of water and solute. Full stop. Will you get mark for this answer? No. This type of a definition you are always to compare. I'm repeatedly telling you, in this type of a solution, in this type of a definition, you are always to compare. What if I talk about isotonic means what? A solution which has got equal amount of water and solute, but in comparison with what? You are comparing with something, so that also has to be written. In comparison with a cell, plant cell, animal cell, whatever. Next, write down hypotonic solution. A solution which has higher concentration of water, higher concentration of water, higher concentration of water, and lower concentration of solutes. Higher concentration of water and lower concentration of solutes as compared to a cell. As compared
lower concentration
of solute
as compared to
as compared to
a cell.
a cell. So what are you writing? This is a solution hypotonic. This has higher amount of water, higher concentration of water and lower concentration of solute. That's the reason the water is moving. Can you write now the definition of hypertonic if you have understood hypotonic? क्या होगा definition? पहले बोलो and then
solution which has higher concentration of solute and lower concentration.
Very good. Higher concentration of solute and lower concentration of water as compared to the cell. That's the reason the water is coming out from the cell. The cell has more water now.
And for hypertonic, what is a solution which has higher concentration?
Solute and lower concentration of water. Remember, in hypertonic, the cell loses water. Higher concentration of solute and lower concentration of water. Higher concentration of solute and lower concentration of water as compared to a cell. Understood? Is this clear?
Sir, solution which has lower concentration to water.
Hypertonic, a solution which has higher concentration of solute and lower concentration of water. That means the sap has more water. That is the reason the water is moving from the cell towards the solution. Or say otherwise, here salt is more, solute is more. That's the reason the cell is trying to give away the water to bring about the equilibrium. Understanding? Read once so you will understand. Now, we are done with this chapter almost. Just few more questions to solve. Let us do some questions. You may take those questions
Just
first.
gave us last question. Last 5 and 6.
Few I gave you. No? Yes, let us try to solve some few more. And you will of course have to go through all the questions. That's chapter 6, right? Diagram drawing, plant and animal cell, you should know. Diagram drawing, plant and animal cell, you should know. Now, I'll tell you, page number 89. Page number 89. Try question number 5 in the textbook itself. Try question number 5 in the textbook itself. No, this is class itself.
Which one?
I said page
89.
Page 89, question number 5. I told you may do it in the textbook.
And you have given two.
Now, that you should be able to do. Then move on to the next page 90. Move on to the next page 90. These are all practical based questions. Let us try one. Number 4. Just read and tell me if you are able to answer this. Number 4.
Page number 90, number 4.
Number 4. Then go to page number 92. Complete number 1. Fill in the blanks. True, false. Match the column. Match the column. Question number 1, 2, 3 from page 92 and 93. 92 and 93. Okay. And then some few questions which you will write in the notebook as homework. First take those questions from very short question 1 mark and 2 marks. What are those questions? Number 2, very short question type. Number 2.
Where to represent what it does.
Correct. Number 6. Number 6. Number 10.
What does it do?
Number 12. Come to 2 mark questions. 17. 21. 22. 26 already I had asked you to do earlier.
Yes, ma'am.
27. 30 in the textbook again. 30 in the textbook again. Okay? And
27.
27 and 30. 27 and 30. Now I will give you some 2 minutes time, 2-3 minutes time. First you make complete number 4. See if you are able to answer. Number 4 and then fill in the blanks, take the straight true words and match the points. See if you are able to do all these. Then those questions which I asked you, the 1 mark and the 2 mark questions which I asked you to do as homework, you will try to write down the question and the answer in the notebook. I want to correct your notebook. Any answer you are not aware, don't write anything. Circle karo, tell me the next class that this is what I am not able to answer now. Then I will give you the answer. But you should learn how to frame your answers. Thoda bhi confusion hai, then leave it. But the ones you are sure, Those only you write and show. Okay. So I will not dictate and give you everything. Many questions are already we have answered. We have already given the answer straight away. But some you should be able to write on your own once the chapter is clear. So 9th standard, any question you will get. See, in the volume we have got hundreds of questions. Will be some 1 or 2 questions you may have not practiced well. How many I gave you? 1 mark, 2 mark. How many questions I gave you?
From 9.
9 questions I gave you. I should have given you more, but because 10 sounds good. 10 questions. Anyways, 1 mark and 2 mark questions only to be written in the notebook. Chalo, first start with number 4. Read number 4. Are you able to understand?
Yes, ma'am.
Yes, from.
What is it? Number 4 says, each of the 3 students ABC were given 5-gram raisins. The raisins were soaked in distilled water at room temperature. Why do we mention distilled water? Because distilled water has got minimum minerals, almost no, you can say. Okay. A soaked the raisins for 10 minutes, B for overnight, C 60 minutes. Then they calculate the percentage of water absorbed. Now answer the following questions. Name the student whose raisins will show the maximum percentage and minimum percentage of water absorbed.
Maximum.
A is 10 minutes, B 6, B is overnight, C is 1 hour, 60 minutes. So which student will have the raisins mostly swollen?
So what will time would I take?
No, now first of all they are only talking about the time period. They are only talking about the time period.
Then B.
Then it is B,
B.
and then C.
C and then A.
No, maximum and minimum. Maximum is B and minimum is A. Okay. Now, if C observes that weight of the raisins has become 7 grams, find the percentage of water absorbed by the raisins. Now, here percentage of water calculation actually you will not get actually the calculation. Okay, but still I will show you the calculation. Now you can read this. You will complete this as homework then. So this also becomes your homework. Fill in the blanks and true, false, match the column. This 3 in the textbook itself, in this book itself. No, don't start writing all of them in the notebook. This, and here itself. And these few questions you will write, writing in the volume. Any question you are not able to answer, not confident, circle karo and send to the next. But at least 4 to 5 questions I am sure you should be able to write. 4 to 5 questions you should be able to write.
ऐसे निकाल यूपीएस चेक कर रही है चक्कर आ रही है
जब हम लिख रही है एंडोस तब तो बोल रहे एंडोस ये मूवमेंट के अगर एंडो बोलते हैं या तो बोल मत
जाता है जल्दी
मुझे भी आता है
बोल क्या करे हाँ पढ़ाता है वाली पढ़ाता है बेटा वाली पढ़ाते हैं वाह